I would like to invite last speaker, second speaker is Dr. Rohit Upadhyay. Dr. Rohit Upadhyay is a researcher scientist at the School of Medicine, Tulane, and was specialized cancer genetics, molecular biology, chemistry engineering, and pharmacology. With the expertise with the bioinformatics, computer, and biology and genetics. So research advanced with the understanding of complexity. So they also serve as a analytical board members and reviews of various educational and national journal contributions to the field of life environment sciences. Over to you.
Hello, can you hear me?
Rohit,
Yes, can you hear me?
yeah, definitely. You can start your presentation.
Okay, thank you so much. Good afternoon, everyone. Today I'm going to talk about my study, mechanisms of Aristolochic acid 1 induced proximal tubule cell injury. So this is the plant Aristolochia clementitis, and this produces aristolochic acid, which is naturally occurring polyaromatic nitrogen compounds. And these are extracted from these plants, which are also known as birthworts or pipe vines, also known as wild ginger. These plants are grown worldwide, used in primarily in the remedies of some folk medicines for arthritis, gout, and inflammation for centuries until they discovered that they have nephrotoxicity and carcinogenicity in upper urothelial tract organs. And then uh, finally, in 70 years ago, there was a Balkan endemic nephropathy, which is also known as BEN, which became recognized after first in Bulgaria, and nobody knew that why it is what it is causing this nephropathy. And then people suspected this plant because this was native plant that area and it was used for some folk medicines. Also, there was one more mycotoxin derived from one specific fungi, and people were suspecting that also. But nobody knew that what is causing this Balkan endemic nephropathy. So basically, these are the countries which synergistically called as Balkan countries. These include Hungary, Romania, Serbia, Bulgaria, and Greece and Italy. So many of these. So uh, as I told before, cause was unknown and suspects were these plant like Aristolochia and microbial metabolites. So initially it was called Chinese herb nephropathy because there were some slimming drugs in Bulgaria which were used mainly by the females, and when they were taking those slimming medicines, they were developing nephrotoxicity, their kidneys were failing, and many of them were having tumors in upper urothelial tissues. And later, people discovered that this was because of the mixture of two different plants. One was Han Fang Zi, and another one was Guang Fang Zi. And because of some botanical classification errors, they were using Aristolochia fangchi instead of using Stephania tetrandra. So it was a erroneous botanical classification. People suspected that this is the cause of it. Later, uh, so basically this uh, Balkan endemic, there was no clear-cut clue what it is causing, like what is the causing factor. So in 1956, when first time it was discovered, people uh, suspected ochratoxins, which are fungal endotoxins, and they evoked aristolochic acid. And it was causing renal interstitial fibrosis and urothelial carcinoma. And later in 1992, people suspected it to be a Chinese herb nephropathy. Later, it was known as aristolochic acid nephropathy from 1996. And again, from 2007, it is known as aristolochic acid and Balkan endemic nephropathy. So basically, it is like a two faces of Janus. This is a proverb said in that region, because in Roman mythology, Janus is a god which has two faces, which looks beginning at the ending, and it shows the transition. So similarly, this Ben endemic, it is also a transition between different suspected plants and endotoxins, but there was no clear-cut knowledge that why it is happening. Then there was one study by Addy they published this paper in 2014. They proposed four pivotal cellular responses in the disease progression. First was the response of tubular epithelial cells to the aggression. So here we can see the tubular injury. And then the second was the loss of endothelial integrity. And third one was interstitial inflammatory responses and the appearance of a new interstitial cell population, the myofibroblasts. So people basically tried to understand what is causing this endemic and what is the mechanism behind it. And also they noticed that the glomeruli was spared. It was mainly in the proximal tubule cells. So proximal tubule cells were the main cells in kidney which were impacted. So coming on the aristolochic acid nephropathy, which is also known as AAN, this causes mainly progressive interstitial nephritis, which leads to the end-stage renal disease. Kidney injury and fibrosis are the predictable features of aristolochic acid nephropathy. It is now used as kidney injury and fibrosis model in vitro and in vivo studies. However, pathophysiology of AAN remains incompletely understood. So as I showed before, this is the plant Aristolochia and this is the compound aristolochic acid. So I'm going to present some of the data which we found in our wet lab and which we published in 2022 in JCMM, and also a couple of more papers in mouse and rats for the urothelial tumor, showing the apoptotic signaling in that. So basically, we used HK2 cells, which are transformed cells of proximal tubules. And we saw that when we treated these cells with aristolochic acid, within 24 hours, we were able to see some morphological differences. Cells were inflamed, their proliferation was less. So we wanted to check what particular dose is impacting this uh, decrease in proliferation. And we checked different doses of aristolochic acid. So basically, 40 micromolar we chose based on the previous studies, and also we saw there was a significant change with this concentration. Also, different time points we checked, and we chose 24 hours. Also, we used different method like BRDUSA, and here you can see in the merged panel that how these red cells are decreasing with time, which shows the proliferation is going down. We also check the viability with the dose response as well as with time response. So viability of these proximal tubule cells was going down with time and dose. We also checked cytotoxicity, dose and time response, similar pattern we found. And then we checked the apoptosis, caspase 3 and 7, because apoptosis is very important mechanism for the aristolochic acid-induced nephropathy. And we saw that 40 micromolar concentration of aristolochic acid is causing apoptosis significantly in these cells. And 24-hour is, again, best time to see this mechanism. Again, we checked these findings with the flow cytometry, and we saw that the cell population increases in aristolochic acid-treated samples in 24 hours. And specifically, we found that the proliferation is going down because of the cell cycle arrest at G2M phase. And then we laid over these two data, which is the cell cycle arrest as well as the apoptotic cells. And we saw that both are synchronized, which shows that this proliferation decrease is happening because of cell cycle arrest at G2M phase. Then we checked the gene expressions specifically for the TLRs and inflammatory cytokines after exposing these proximal tubule cells with aristolochic acid. So toll-like receptors are pattern recognition receptors, also known as PRRs. They have a unique and essential function in innate immunity. Basically, there are 10 known TLRs in humans, which could be on cell surface like TLR 1, 2, 4, 5, or 6. And some TLRs are present on the membranes of endosomes or lysosomes like TLR 3, 7, and 9. So in our experiment, we found that NLRP3, which is involved in the inflammasome, and some particular TLRs like TLR 4, 2, 6, but specifically TLR 9 were overexpressed in aristolochic acid treated proximal tubule cells. We again checked these findings with the immunofluorescence, and we saw LCN2, which is a kidney injury marker, by the way. This is a neutrophil marker of kidney injury, which was overexpressed in AA-treated cells, and TLR9 was also overexpressed. So TLR9 is of particular interest because this is basically of alien origin generally, which is from the viral or fungal origin. But in some cases, it can be endogenous also in origin, mainly when there is a mitochondrial uh, dysfunction, then mitochondrial DNA comes out from the mitochondria and it attaches to the TLR9. So TLR9 basically is a ligand for the mitochondrial DNA. So basically, mitochondrial DNA, they can work as a damp molecules to activate TLR9. So then we checked ROS production because when TLRs are activated, they can contribute in the ROS production, and we checked ROS in AA-treated proximal tubules, and we saw that ROS production is significantly upregulated in these cells in comparison to the positive control of hydrogen peroxide. And when we use Tempol, which is a ROS inhibitor, then it decreased ROS production in HK2 cells, which makes sense. And then similar to ROS, NOS is also important. These are nitrogen oxygen species. And we found that inducible NOS was increased in aristolochic acid treated cells. And NO production, we also checked the nitric oxide production in these cells, and we saw that nitric oxide production was also increased. So basically, it was not only the ROS, but NOS was also involved. So because ROS was increased and we saw that TLR9 was upregulated, so main suspect was now mitochondrial DNA, because that is a DAMP molecule which mimics the alien origin of DAMPs, or like mainly the PAMP molecules. Those are the pathogen-derived um, molecules. But in the cell culture, there is no alien um, endotoxin or something. So only thing which could activate TLR9 may be mitochondrial DNA. So therefore, we checked mitochondrial dysfunction by checking the membrane potential of these cells. So we used a specific assay. This is known as TEMRI assay, or tetramethyl rhodamine ethyl ester assay. And we again found that with the dose-dependent manner, aristolochic acid is decreasing the membrane potential of mitochondria. So by the histopathology of these patients, we know that there is a fibrosis in tubular cells when there is this aristolochic acid-induced nephropathy. Therefore, we checked alpha SMA, which is a known marker of fibrosis, and it should go up when there is a fibrosis. And here we can see that alpha SMA, which is a green fluorescent, here you can see it is increased in aristolochic acid-treated cells in comparison to the controls. Another marker we used E-cadherin. E-cadherin should go down when there is a fibrosis, and we found similar pattern here. E-cadherin, which is again labeled with a green color, and it is going down in aristolochic acid-treated cells. Internal control is phalloidin, which is in red. So basically now we knew that what is happening in these proximal tubule cells, there are ROS production and NOS production, which is activating TLRs, specifically TLR9, which is decreasing membrane potential of mitochondria, and mitochondrial DNA is coming out. So to inhibit this process, or to ameliorate this injury, we tried to use Tempol and TLR9 siRNA. So in this panel, if you see the second, the number 2, it is showing aristolochic acid-treated cells. And then 5 and 6 are aristolochic acid with Tempol and TLR9 respectively. So we treated these cells with Tempol, pre-treated with Tempol, and we silenced TLR9. And then we treated with the aristolochic acid, and we saw this change in gene expression, as you can see in this heat plot. So basically, it was rescuing cells from injury. LCN2 was decreasing, which is a known kidney injury marker. And most of those markers which were upregulated in aristolochic acid-treated cells now were those were compared to aristolochic acid, those were normal now. Again, we checked apoptosis, because apoptosis is the main morphological change or phenotypic change what we see in this nephropathy. So in aristolochic acid, if you see this cleaved caspase 3 is upregulated, while when we silenced TLR9, it was decreased, and similarly in Tempol pre-treated cells as well. Again, in this experiment, we saw that when we use Tempol and TLR9 siRNA, they decreased mitochondrial dysfunction as well. As you can see, this Tempol is significantly upregulating this effect from the aristolochic acid. So basically, in summary, if we talk about these all experiments, then aristolochic acid, it goes into the proximal tubule cell, increases the ROS, and this ROS attacks to the mitochondria, and it decreases its membrane potential, which in turn takes out its DNA, which acts as a damp molecule, which attaches to the TLR9, or TLR9 acts as its ligand. Also, there is involvement of TLR3 and some other damp molecules like HMGB1, which is a ligand for TLR2 and 4, but most specifically TLR9. And it causes kidney injury, which is showed by the LCN2 overexpression. And this all causes the inflammation. And that's why we saw the inflammatory markers upregulation like TNF alpha, IL-6, IL-18, TGF beta, IL-1 beta, and NLRP3. And when all this inflammation is happening and cell is under injury, it shows decrease in proliferation, viability, increase in cytotoxicity, apoptosis, and cell cycle arrest, while mitochondrial membrane potential goes down. And also we saw that alpha SMA goes up and E-cadherin goes down for the fibrosis. While when we use Tempol to inhibit ROS, it rescued these cells from the injury. Similarly, TLR9 siRNA, it inhibited further inflammation in these cells and again rescued these proximal tubule cells. So in my conclusion of this study, it confirms that aristolochic acid 1 is a potent nephrotoxin, primarily affecting proximal tubule cells, and extensive fibrosis is a hallmark of aristolochic acid nephrotoxicity. And we can, by this study, we knew that TLR9 has a prominent role in it, and which indicates role of innate immunity in aristolochic acid-induced fibrogenesis in the kidney. Also, it shows the important role of oxygen radicals, damp molecules, TLR activation, and we can ameliorate all these effects by ROS inhibitor and TLR9 siRNA. However, this is the mechanism we know, but still, there are so many new cases of aristolochic acid nephropathy throughout the world, despite there are some guidelines from FDA showing the warnings regarding the safety of botanical remedies containing aristolochic acid and plants containing aristolochic acid. But still these drugs, medicines, as a folk medicines for slimming drug, for arthritis treatment drugs, still these medicines which are not properly regulated, they have aristolochic acid in them. So we have to be careful while we are choosing any medicine which has this compound in it. So these are my acknowledgements. These studies were done in collaboration of Imperial College of London and my institute, Tulane University. And I'm open for questions. Thank you.
Thank you, Dr. Rohit. It was a wonderful presentation. So now the session is open for the questions. Yeah, thank you, Dr. Rohit.
Thank you so much for having me.
Yeah.
Thank you, sir, for the energetic lecture with a wonderful time. For all this speakers sharing their knowledge and groundbreaking research and to all interested participants for engaging and throughout improving. With this, we conclude presentation of technical 5. Then we have a big round of applause for all the